Students, let us see the molecular structure of DNA. Molecular structure of DNA is having the primary structure called covalent backbone structure, which is having a repeated deoxyribose connected by phosphodiester bond, and the polarity is 5' to 3' because the 5' end is having a free phosphate and 3' it is having a free OH group. And the DNA single strand, when it joined with another strand, it formed double helical structure. Following the Watson and Crick model, Charkov's rule, it is forming a base pairing. 10 bases per turn is there, and it is forming a minor groove and major groove. The minor groove is very shallow groove, and major groove is a deep groove. Minor groove is the place where the histone proteins bind. Major groove is the place where all the regulatory proteins will bind. And this structure is stabilized by hydrogen bonds. And as per the Watson and Crick model, base pairing happens, purines are equal to pyrimidines. It is also stabilized by base stacking hydrophobic interaction between the adjacent bases. This DNA will be having different forms depending on the environment in the cell. If the humidity goes down below 75 percentage, it forms A form of DNA where it is right-handed helix and 11 base pair per turn. If it is the natural form at which is stable at 92% humidity in the cell, follow the Watson and Crick model having 10 base per turn. Whereas one more form is there which is very rare, transient form, 12 base pair per turn. This is not having differentiation between major and minor groove, having single groove, left-handed, and it is having zigzag arrangement. In addition to these forms, in between, if it is having a palindromic sequence, it will form a hairpin-like structure in that the bases which are not complementary to each other will form a loop. So hairpin and loop structure will be formed. If it formed in both the strand, then it is cruciform structure. In addition, sometimes the mirror image may leads to the complementarity sequence which will turn and form one more base pairing apart from this Watson and Crick base pairing existing. So this is not following Watson and Crick base pairing, and the third strand is giving a triplet structure called Hoxton bend or Hoxton pair or HDNA. Sometimes the fourth strand also can base pair and it will lead to the tetraplet structure. E. coli genome is also forming a scaffold with proteins, and the DNA of both prokaryotes and eukaryotes, they can form the quaternary structure when it is complexed with the protein like histone or other proteins in prokaryotes, or even the higher order of folding, it will give the quaternary structure. Similarly, RNA is also having a primary structure, covalent backbone structure, where the instead of deoxyribose, we have a ribose, which is having OH in the second carbon, and the structure is similar to the DNA strand, which is single-stranded. There are three different types of RNA, and here the mRNA is linear RNA, but if it is having a complementarity sequence, a self-complementarity hairpin loop structure can be formed. Whereas in tRNA, it is folded to form the cloverleaf structure, and rRNA, it will be having more of self-complementary foldings. The mRNA, we have plenty for each protein, one mRNA is there. And tRNA, for each amino acid, we have one tRNA. So 20 will be there. In addition, some will be having, some organisms will be having more number of tRNA. Maximum 10, 50 tRNA can be there. rRNA in prokaryotes, three classes, 5s, 16s, and 23s. Eukaryotes, we have in the nucleolus three types, and outside nucleolus one type. It is also having secondary and tertiary structure because of the self-complementary folding base pairing. The higher order of folding is tertiary, as you see in the tRNA. And secondary structure is simple cloverleaf structure. This RNA also can knot itself more of RNAs. It will form a complex structure, quaternary structure, example aptamer, or else it can bind with the protein and form a complex such as RNPs and then ribosome, spliceosome, so on. They are the examples for the quaternary structure of RNA. The last part is bacterial recombination. Bacterial recombination is the one where the DNA is come closer and then the exchange happens. So this can be done in prokaryotes by three process. One is transformation, next is transduction, next is conjugation. Transformation, here the DNA adhere to the cell from the surrounding a naked DNA, and the double helical structure cannot enter. Only single strand can enter it into the cell, and the cell should be competent to take this, and it will allow the entry of single strand DNA of having maximum of one or two genes, not more than. Only that much single stranded DNA can enter into the cell. And that single strand is stabilized by single strand binding protein, and recombinase protein will help for exchange of the chromosome. And how this exchange happens, the general mechanism of recombination is explained by different models. Here, one model is shown where the incoming gene DNA fragment can invade into the existing chromosomal DNA like this, and it will form a crisscross structure, and that joints will be then cleaved by the endonuclease enzyme, and the exchange happens like this. And D loop or displacement loop method is followed to complete the replication and close it to form the exchange. If the cell is not competent, especially E. coli is not competent to take any DNA, but we need to use the E. coli for transformation purpose, for genetic engineering purpose, what we have to do is increase the temperature, and so that the pore size will be increased, and the DNA, the plasmid or any other external DNA negatively charged should come closer to the membrane. Membrane is having negative charge. That will be neutralized by addition of chemical calcium chloride, and then it can enter. Similarly, the electric pulse also can cause this pore, and that process is called electroporation. Chemical or electric pulse can facilitate such entry of DNA in non-competent cells. Now, transduction is the next topic or recombination method. Here, the virus will carry the DNA, which will be injected into the cell, and recombination is facilitated. There are two types. Generalized transduction, it is done by lytic life cycle, and this lytic virus infect the donor bacterium, and it multiply itself, then break down the chromosome into pieces. If the pieces if the pieces are having the gene, that will be carried by some of the virus, and that virus is called as generalized transducer. Then when it infect the recipient cell, it will leave the DNA and exchange happens. The phages which undergo such transduction are shown here, and some are not carrying out this transduction. The phages, they completely degrade the DNA, cannot take any DNA, and that do not degrade also cannot take any DNA. And then the some phages are very specific; they will pack only viral gene, and they also cannot undergo such transduction. And then specialized transduction, it occurs only in temperature or lysogenic virus. It is done by virus, and that will infect the donor cell, integrate into the specific site between gal and biogene, and this integration can stay until an infection comes. Once infection comes, this virus, it try to come out. When it come out, some in the frequency of 1 per 10 the power 6, they will take one gene gal or bio along with it by leaving its replication gene into the chromosome. So it becomes replication defective. virus. So this can infect the recipient cell. There also it is integrated between gal and bio only, but now it is replication defective. It cannot replicate on its own. It will replicate along with the chromosome. In order to increase the frequency, a secondary infection with the wild type lambda fetch that will help for the multiplication, or else the gal plus can exchange with the gal minus. This is called high frequency transduction. And conjugation, it will form a conjugation tube and a copy of the plasmid will be entering into the recipient. This is not causing recombination, only the plasmid is going. We have a HFR condition where the plasmid integrate into the chromosome, and in that condition, when it undergo recombination, at the end of the plasmid, nick happens, and the chromosomal DNA of 1, 2, or 3 will enter into the recipient cell. There, the genetic exchange takes place. And the plasmid, if it is integrated into the chromosome, it can be excised out. When it is excised out, some will be excised out by carrying few genes from the recipient chromosome. They are called F prime factor. When they goes to the recipient, this gene which is taken from the donor will be then multiplied by self replication, because F prime is also able to replicate on its own like F plasmid. Thank you.